Thank you. Thank you for making the time to come out on a Saturday to learn with all of us about Parkinson's disease and everything that's going on in the world of Parkinson's. So right now I'm going to go through a somewhat brief tour of what's new in the world of Parkinson's in terms of drug development and treatment development. So standard side, no relevant disclosures here. So here, I don't know how much you can see of that screen, but this is a timeline of the developments that have gone on since Parkinson's was first described systematically in 1817, so almost 200 years ago. And as you can see, over the last 200 years, a lot has happened in terms of how we understand the disease and in terms of what treatments have come up. Right? About 50, 60 years ago or so, Levodopa was first produced and became commercially available and has been the standard of treatment for Parkinson's for really that length of time. And since that time, right, even though it takes up a lot of space on that screen behind me, the timeline has been quite short. The intervals between drug developments, between developments in understanding the disease has gotten shorter and shorter, right, year after year. There's more and more stuff that's happening. And so we're in an exciting time right now where this will likely continue to occur, where we understand more and more about the disease and more treatments are developed year after year. Now keep in mind though, every time a drug is developed, it does take some time. Right? And for many of us who have watched drug development over the years, on average, any drug will take about 10 years or so from when it's discovered or created to when it's actually approved by the FDA for clinical and human use. Right, so throughout today, we're going to be talking about different drugs and different medications at different stages of development and research. And this is just to kind of give you a sense of what actually happens. Right, so initially, drugs have to be discovered in a lab or tested on animals and so on. That's typically the preclinical phase. Right? Then it goes through multiple phases of clinical testing in people. So phase 1 studies, phase 2 studies, phase 3 studies, before that data can then be submitted to a regulatory agency like the FDA for approval. And just to give you a sense, phase 1 studies are typically small-scale studies, not very many people involved, but it's primarily to look at the safety of a medication for human use. When you go to phase 2, you bring in some more people to study that medication, and then you're looking more at safety and how does this drug actually work? Is it providing the benefit that we want it to? And then the phase 3 studies are really when you bring in a large group of people with the disease in question and you test the drug, again looking at safety, but now also looking at a broader scale efficacy. Is the drug doing what we think it's supposed to be doing? And so we're going to be seeing a lot of different medications that are going through these stages throughout the next half hour. So Levodopa. Everyone is familiar with Levodopa. Levodopa is the gold standard medication for the treatment of Parkinson's disease. Right? And the reason why, and we've talked a bit about this today, is that Parkinson's, as we understand it, is a disease where the brain does not make enough of a chemical called dopamine. And so the way we treat Parkinson's disease is to give the brain the dopamine that it cannot make on its own in the form of a pill. And Levodopa is the most direct way of doing that. Right? And because of that, a lot of drugs have come along that play on different variations of Levodopa, different formulations that try to extend the duration of its effect to minimize the side effects of Levodopa. Right? And you have things like Parcopa, Sitamet CR, Rytari. Right? These are all medications that are playing on the same idea. They're still Levodopa, just formulated in a different way to act a bit differently. We heard in the last half hour, for those of you who were in this room, that timing is everything, right? And that's a theme of Parkinson's disease that carries through and through, right? So here's a graph to show you kind of what happens with Levodopa in the body once you start taking the doses. And understanding this phenomenon is important for a few things, right? Number one, it's actually how Levodopa works, and so understanding it helps you take care of yourself better. But number two, it's a core principle of drug development in that people are looking at ways to provide Levodopa in a way that extends its effect and minimizes side effects. Right? So on the y-axis there, you have dopamine levels inside the body, and the x-axis is time over the course of the day, starting from the morning all the way to the evening. Right? And when you take your first dose of Levodopa, right? The levodopa goes up, and then it goes down, then it goes up, and then it goes down. And for that reason, you take multiple doses of levodopa throughout the day to maintain a reasonable level of levodopa in the body and maintain a reasonable level of effect. You essentially want to be within some zone of consistent levodopa dosing, right, to avoid wearing off of the levodopa and to avoid side effects, right, of which could be nausea, dyskinesias, hallucinations, what have you. And as people get older with Parkinson's disease, the duration to which levodopa lasts, it shortens, and the frequency of wearing off increases, right? So for drug development, the key things here are how do we get levodopa to last longer, and how do we avoid the side effects by avoiding those peaks and valleys of levodopa throughout the day. So we're going to start talking about subcutaneous infusion therapies. So the idea of these therapies are So subcutaneous infusion therapies, the idea of these therapies are how to provide a way to have a continuous, sustained flow of medication in the body to avoid those peaks and valleys. We're going to start this conversation by talking about apomorphine, which is not quite levodopa, but is a dopamine agonist. So it acts on the dopamine receptors without actually being a dopamine chemical or molecule itself. Morphine is in the name, but has nothing to do with opioids. It's a breakdown product of opioids, and so that's kind of why it's called that. But essentially, it's a dopamine agonist that has been used in a subcutaneous fashion. Historically, not so much continuously in the US, but as needed therapy. So currently it's available as subcutaneous injections, marketed as Apokin, and this is for people who have sudden wearing off but they need a quick response. So you essentially just inject it under the skin, and it provides you with an on effect. Kinmobi, which is a sublingual formulation of apomorphine, was available, but discontinued in the past year because it wasn't being used as much, has the same principle, right? Something that you use for when levodopa wears off. Right? The reason why I'm talking about apomorphine like this right now is that apomorphine does have a continuous infusion formulation. And essentially the way it works is it's very similar to an insulin pump for people with type 1 diabetes, right? So in people with diabetes, they don't make insulin, so you give them insulin by pump. And so the idea for Parkinson's disease, it would be very similar. You provide the medication through a pumping mechanism that provides it in a sustained way throughout the day. So again, trying to avoid those peaks and valleys of ons and offs. And these infusions of apomorphine have been shown to increase on time with less troublesome dyskinesias and less side effects, and reduce off time overall on the order of anywhere between 2 to 3 hours per day. What's interesting about apomorphine is it has actually been available in the world for quite some time. It was approved in Europe in the 1990s, so well over 20 years ago, but it hasn't quite made its way to North America yet. So the company that makes this drug has refiled a new drug application with the FDA, and the decision is expected sometime soon. I was actually talking to Dr. Ramirez Zamora in the back there about this a couple of days ago, and I think the expected announcement day was yesterday, but we haven't heard anything yet. So we're going to hang tight, and it should be announced fairly soon, hopefully. So let's go back to levodopa, right? So apomorphine is not quite levodopa, it's a dopamine agonist, right? Then the question is, well, can we actually get levodopa into the body in a continuous fashion? And here's where this product, ABB951, also known as phoslevodopa and phoscarbidopa, come in, right? So it's not quite levodopa, it's phoslevodopa, and it is what we call a prodrug of levodopa. So on its own, it is inactive, it doesn't do anything, but once it gets into the body, it gets converted into levodopa, which then takes its effect, right? And the studies for phoslevodopa have shown a very similar effect to apomorphine, in that when you infuse this drug through a pump under the skin, it increases on times, right, for the day, and it decreases off time. So again, avoiding those peaks and valleys. Now, what's interesting about this one is that it is a fairly recent development, right? So in the last year, 2023, it was approved in Canada, the UK, and the European Union for use. And as of this year, 2024, we're starting to see people who are getting this device, getting this pump, and getting this therapy. So it'll be really interesting to see, really over the next year, next 2 years, how this plays out in real life as we see more and more of this come online. Another drug that's, so in the US, it right now is undergoing FDA review. And so in March of last year, the FDA had asked for more information regarding not necessarily the drug, but actually the pumping mechanism to deliver the drug. So we know it's in the pipeline somewhere. So phospholevodopa is a therapy to be looking out for in the very near future. A similar concept here is a drug called ND0612. And essentially, it's a liquid formulation of levodopa. Because you can imagine right now, levodopa, for the most part, is a solid medication. It's in pill form, it's in gel form, it's in the phospholevodopa form. But there hasn't really been a usable liquid infusion. And so this ND0612 is a liquid formulation of levodopa that, again, would be infused subcutaneously under the skin via a pump. Right? So similar concept. And again, it's showing very similar results. So this is very fresh off the press. On March 15th, 2024, this is only 3 weeks ago, the results of a phase 3 clinical trial were released, and it showed that, very similar to the phospholevodopa and the apomorphine, that it does increase on times throughout the day with less troublesome dyskinesias and side effects, with less off times. So again, on the order of giving someone up to 2 hours or so more on time throughout the day. So those are the subcutaneous medications, and that's kind of where a lot of the excitement is right now, because it does help people with Parkinson's avoid some of the complications of advancing disease, namely wearing off and motor fluctuations. There is a medication that's in the pipelines called IPX203, and this is a different way of packaging the typical levodopa-carbidopa, or carbidopa-levodopa, in an extended release form, right? So currently the extended release formulations are Cinemet CR, right, or Rytari, and this is a play off that as well, right? So having a component of immediate release and having a component of extended release and packaging them together to give someone more on time, less off time. And so the results of that study were just published last year, and that is still in the pipelines. I'm going to shift gears a little bit here. So what I just talked about are the very soon-to-be upcoming medications and therapies. But the bigger question of Parkinson's disease is, well, how do we actually fix the problem of Parkinson's? How do we cure the disease? And to get at that, we have to kind of understand, well, why does Parkinson's actually happen in the first place? The short answer right now is we don't really know. But the longer answer has to do with this figure there with very small text. And what it's showing there really is that there's a lot of stuff happening inside a cell, inside your body, inside the brain, to cause the disease to possibly occur, right? So essentially, there's a lot of things that can go on in Parkinson's disease, or in a brain to cause Parkinson's disease. Alpha-synuclein has been talked about a lot, right? And alpha-synuclein is a protein that then accumulates and creates Lewy bodies, and that's thought to create some kind of damage to the neurons or brain cells that then prompt Parkinson's disease to develop. There's a lot of things that can go into that too, right? There could be mitochondrial dysfunction. Mitochondria are the powerhouses, the energy-producing factories of a cell. If those are dysfunctional, they can cause cell damage, cell death, and that can be related to oxidative stress, right? So basically, we're kind of seeing here, right, mitochondria here, oxidative stress, that can contribute to the development of pathology in the brain. You have things like the lysosomes, or essentially metabolic garbage reducing parts of the cell, right? If toxins accumulate, then the cell can be damaged, et cetera. So there's a lot of stuff going on inside the brain that could possibly lead to someone having Parkinson's disease. And researchers are looking at all of these different components to sort out, well, what are the actual mechanisms of the disease being produced, and how can we address those mechanisms to either stop the disease from progressing or cure the disease. So these understanding why Parkinson's develops helps us create approaches to curative treatment. And the approaches include, number one, stopping disease progression. And this is done by addressing the potential mechanisms of disease. Neuroprotection, right, in terms of protecting what is still intact in the brain, what is not damaged, ensuring that those functional parts of the brain and body do not degenerate. And number three, how do we restore what is lost? Right? So by the time someone with Parkinson's develops symptoms, the part of the brain that we always talk about, which is the substantia nigra, it's already lost up to 70% of its neurons. So how do we give those neurons and cells back to the body in a functionally meaningful way? Currently, right, there aren't any real medications that can do any of these three things, and the ideal medication would do all three. But that is an area of heavy research to understand how can we actually achieve those goals. Now, the medications I will talk about in the next little bit, none of them are approved whatsoever for Parkinson's disease. Some of the medications are very experimental. They're new chemical developments. Others are repurposed medications, so medications that are used for other diseases and are being repurposed for Parkinson's disease. Do not try any of these on your own because, again, they're not approved for Parkinson's disease use. Okay, so we talked about alpha-synuclein, right? That protein that aggregates, that builds up in neurons and is thought to cause neuronal damage. An easy thought would be, if we block those chemicals, those balls of alpha-synuclein, can we prevent the disease from happening, right? So monoclonal antibodies idea here were that antibodies would attach to the alpha-synuclein and promote clearance of the alpha-synuclein. So your body would get rid of these bad molecules and thereby perhaps prevent Parkinson's from getting worse. So there were two studies that were done, and the results came out last year. There was a lot of hope with these two. Sapanimab and prasinezumab. Unfortunately, there was no difference from placebo between using the infusions of these antibodies versus a placebo. So that's still being looked at. Of course, this begs the question of, well, how much is alpha-synuclein truly impacting the brain? How much is it truly contributing to Parkinson's disease? And that is an area of huge discussion and debate in the research world right now. Another approach to addressing alpha-synuclein is what people have called a vaccination approach. So to prime the body to recognize these alpha-synuclein molecules and to clear them and get rid of them by using the body's immune system. So one drug that's being looked at right now is called PD01A. I know I'm saying a lot of letters and numbers here. And this is designed as a small protein that is supposed to trigger the body's immune system to recognize it as being a foreign body and to essentially attack it and get rid of it. And so the studies that have been done, right, show that it is safe to use in humans and that it does cause antibodies to alpha-synuclein to increase in the body. So it does seem to actually cause an immune response to alpha-synuclein. And in this one study here, it showed that when you measure the levels of alpha-synuclein in the cerebrospinal fluid, that's the fluid that bathes the brain and spinal cord, that it does actually decrease the amount of alpha-synuclein there. So perhaps this vaccination approach has some merit, and that it could actually help with the clearance and elimination of alpha-synuclein, which may be bad for the brain. And so right now, phase 2 trials are in progress, so this is again ongoing in terms of its research. Another chemical, UB312, also goes and tries to use this vaccination approach. And the last one was a subcutaneous injection under the skin. This is an intramuscular injection just in the muscle. So again, very much like standard vaccinations. And again, the idea here is you're mimicking the alpha-synuclein so that the body can recognize it as abnormal. And the initial studies have shown that it is safe, and it does cause the body to produce antibodies. So another promising therapy to be looking at. And so right now, so the first study was done in people who did not have Parkinson's disease, and it showed that those people who were otherwise healthy were able to produce these antibodies. Another study is now going on looking at people with Parkinson's to see that, whether it's, again, safe and does it produce the response that we'd expect it to. So shifting gears here a little bit, other approaches to drug development go after genetic mechanisms of Parkinson's disease. So GBA, or glucocerebrosidase, is one of those methods. The GBA1 gene, which produces this chemical called glucocerebrosidase, or GBA, has been associated with Parkinson's disease for quite some time now. And variants in the gene, or mutations in the gene, are one of the most common risk factors for someone to develop Parkinson's disease. And if someone has an abnormality in this gene, what is thought to happen is that it causes the protein to act abnormally, to become less effective than it should be, and it's then thought to lead to aggregation and clumping of alpha-synuclein, which then leads to pathology and Parkinson's disease. So the thinking here is if the GBA gene isn't working the way it's supposed to, and you're not creating enough of this enzyme, can we replace this enzyme? Can we cause a boost in its activity to then help treat Parkinson's? And one of the chemicals that are being looked at right now is called ambroxol, and this medication has been used for quite some time, mostly in the world of pulmonology and respirology, looking at phlegm and mucus clearance in respiratory disease. So this drug is available, but it is not tested or approved for Parkinson's disease outside of these studies. But these studies have shown that the ambroxol can increase the levels of GBA protein, GBA enzyme. And so if it does do that, right, can it cause the levels to be high enough or to work well enough to help with alpha-synuclein clearance and prevention of development of Parkinson's? So ongoing trials are planned and going on towards phase 3. So another gene that's implicated or associated with Parkinson's disease is what we call the LRRK2 gene, and it encodes for the leucine-rich repeat kinase, so another enzyme in the body. And variants or mutations of this gene would cause the most common form of genetic Parkinson's disease. So we do know that it does have some mechanistic relevance to Parkinson's. But what's interesting with these patients, right, who do have LRRK2 mutations is that they often don't have a lot of alpha-synuclein in their brain on pathology once the brains are looked at in autopsy. So is this the same disease as what we've been talking about, or is it a different flavor of Parkinson's? We don't quite know yet. And so the idea here is if there is a mutation in this LRRK2 gene, there's thought to be an enhancement of the enzyme activity. So it's working more than it should be. And by doing that, it's thought to cause what we call a toxic gain of function, that it's doing more than it's supposed to, and it's actually causing toxicity to the brain, and then cell damage and the development of disease. So drugs and drug developers are looking at how do we inhibit LRRK2, right, to help prevent and treat Parkinson's. So there's a chemical called BIIB122, or DNL151. And this has been tried and studied, and in this trial has been shown to decrease LRRK2 levels. So again, showing what we think is an expected effect and providing promise that it could be a Parkinson's disease drug. Right now it's gone on to a phase 2 trial. There was a phase 3 trial as well, but the companies decided that it was going to take way too long, that the timeline for completion of this was going to be, I think it was like 2030, so 6 more years from now, and they decided to consolidate their resources and go with a higher quality, faster study to kind of get more information out there, but one chemical to watch out for. Another mechanism of Parkinson's disease is mitochondrial dysfunction, right? Again, mitochondria are the powerhouses, the energy houses of the cell. If you don't make enough energy, cells unfortunately die. If the mitochondria don't work properly, you cause oxidative stress, which again damages cells and causes things to die. So the idea here is, can we treat mitochondrial dysfunction by helping the mitochondria work a bit better, work more efficiently, and can that help with treatment? So there is a chemical called urodeoxycholic acid, or UCDA for short. This is a chemical that's, or a drug really, that's used to treat cholangitis. So it is a naturally occurring bile acid. And it's been shown to make these mitochondria in the body work a bit more efficiently. And so with that thinking, it's been tried for Parkinson's disease as well. So a phase 2 trial published results last year and showed that the chemical was quite safe to use in humans, and we're still kind of awaiting more information on how does it actually impact the progression and change of Parkinson's disease over time. I'm going to shift gears again. So another mechanism is looking at how does the cell or a neuron metabolize waste products, how does it kind of do its day-to-day job, and there's a chemical called c-Abl, or Abelson tyrosine kinase, and this is generally an indicator of oxidative stress, right? So oxidative stress in a cell is bad. If you have oxidative stress, it can cause damage, it can cause cell death, and lead to disease. So the idea here is, can we do something or inhibit c-Abl to then allow the body, allow the brain to function as it should? And there's a chemical called nilotinib that has been investigated, and they've already completed 2 phase 2 trials of this medication. Unfortunately, though, it showed very limited clinical effect in terms of efficacy in people with Parkinson's disease. But the question here was, well, is it because the chemical doesn't do anything at all, or is it because the chemical didn't actually make its way into the brain, right, into the nervous system to actually take its effect? Right, so as a result, there are other c-Abl inhibitors that are currently being investigated to see if there's any indication that they could be useful for the treatment of Parkinson's. Neuroinflammation has been a big topic, right? So neuroinflammation, inflammation of the brain, inflammation of the gut, inflammation in general would cause things to not work properly, can cause cell damage, can cause cell death, and then disease development, right? So there is this question of, if we address inflammation, can we settle down Parkinson's or help slow its progression? There are a lot of chemicals that are being investigated for this purpose, many of which are already commercially available for other things. The big one would be azathioprine, which is a very well-known and well-used immunomodulator, and that's being repurposed and looked at for Parkinson's disease. And it's just an example of one of many drugs that are being looked at in this way. This has been a big topic, so glucagon-like 1 agonists. And that long name might not mean a whole lot to you, but this class of drugs, right, has been approved and widely used for diabetes and weight management. If you've heard of Ozempic or its cousins, this is the class of medications, right? So in the general population, it is being widely used. The question here is, well, does it have a neuroprotective effect in Parkinson's? Can it help prevent further damage to neurons that are still there, that are still functioning in an appropriate way? And when you look at mice and rodents, the studies seem to say, it could protect neurons from further damage and prevent Parkinson's disease. The unfortunate part is that mice and rodents don't quite get Parkinson's the same way that we do. And so because of that, we have to look at this in humans and say, well, is it actually helpful? And in recent years, there have been several big trials, including one that just where the results came out this past week, that looked at 2 different GLP-1 agonists, so exenatide and lexanatide, and they both kind of showed similar things, right? These were medications that were tested in people over the period of about a year, and essentially they found that there was a difference, right? So that people who took these drugs had better motor scores in their Parkinson's disease than people who took a placebo. Now, the catch here is that the effect of the difference was quite small. So the question, of course, is, well, yes, these drugs maybe have some benefit, but is that benefit clinically meaningful? And I think that's a question that we will have to answer over the next, in the near future, to get a better understanding of what these medications can do for people with Parkinson's. So the important things to know about are the side effects. And the side effects are the same, essentially, as for people who do not have Parkinson's and use these medications. So nausea, decreased appetite, weight loss, and all of these things are of importance to people with Parkinson's, right? Because these are the side effects you don't necessarily want, and can also just make your life worse. Those two medications are some of the ones that have been tried, but others are also being looked at. Right? There's many medications in this class, and of course the big one is Ozempic or semaglutide. So again, widely available, but not approved for use in Parkinson's. Stem cells have also been a big topic. We get asked about stem cells quite often, and the idea, it's very attractive because in Parkinson's you have cells that are not there anymore, so can you replace the cells by using stem cells to help regrow dopamine neurons or other neurons that are no longer there in Parkinson's? For the most part, all stem cell studies are in the early stages of investigation, so there's nothing that's close to clinical use. But it's a hot topic, and it's something to keep an eye on. There's many groups around the world, as is shown by this map, that are working on this problem. But we need to keep learning more about, well, I guess, one, how do we actually inject stem cells into someone? Are we getting them to where they need to be? And are they doing what we want them to do? These are unanswered questions that we'll hopefully have more answers to over the coming years. So shifting gears again, so new knowledge is really important in terms of how Parkinson's disease works. New knowledge leads to new understanding, and new understanding then hopefully leads to new therapies. So I've spent a lot of time talking about what's in the pipelines, what's being investigated for upcoming therapies. I want to shift a little bit now to talk about, well, how do we create new understanding to know where to target our drug development? How do we understand the disease better to create new drugs and therapies? So biomarkers is a huge topic, and the Michael J. Fox Foundation, among other national, international organizations, are funding projects related to looking at biomarkers that indicate a disease state, right? Can we use something in the blood or the cerebrospinal fluid or in saliva to get a sense of, does someone have Parkinson's disease, what stage that Parkinson's disease is in, and how do we use that to treat disease? There's also imaging biomarkers. So I thought this was kind of interesting, but we don't really talk a lot about it, is looking at neuromelanin deposits in the brain. So neuromelanin is normal in people's brains, and it does develop and accumulate in the substantia nigra where there's disease in Parkinson's. So you can see over here, right on the top is a normal brain where you have a lot of bright stuff showing neuromelanin. The bottom is a Parkinson's brain, and you see a decrease in that. So this could be an indicator of Parkinson's disease and help people understand where the disease is at and how to treat it. Genetics is also a big topic, and so we already heard a bit from Dr. Malati earlier about the PED generation study that's being done through the Parkinson's Foundation, and we did talk in the last half hour about how certain genes are associated with the disease and how they can cause disease. So better understanding what the genetics of the disease are, the better we can help create and use more specific targeted therapies. And the big thing in the last year, 2023, was artificial intelligence. And this is a big question mark, right? Well, everyone's talking about AI for everything, so why not AI for Parkinson's disease as well? And I think we'll, again, learn more about it as things develop, but the way things look, artificial intelligence can be used for a few things, right? Number one, for research purposes, to do deep dives into big data and to help analyze data to create new knowledge, create new understanding. And in the clinical world, right, it may be helpful to use AI for things like monitoring disease, diagnostics, and response to treatment. So a lot of things kind of coming up in this area, but we'll see really over the next few years. So to summarize, we've gone through a lot here, but number one, we talked about upcoming therapies. And really, in my mind, those are the subcutaneous infusion therapies that are really very much on the horizon for their use. Future therapies, so looking at a variety of drugs, right, either new chemicals or repurposed drugs, targeting specific disease mechanisms to treat Parkinson's, and looking at number three, areas of investigation. How do we, again, create new knowledge and understanding to create new therapies? So thank you very much for your attention, and glad that you're all here today.